Welcome, everybody, and it's a distinct pleasure to introduce our lunchtime entertainment for the meeting. I've known actually David Newman for many years, ever since I was a graduate student in the Scheuer Lab, and I just have two things that I want to mention about David's actually very colorful history that are very important to the field of natural products. And the first is that he served as the chief of the natural products branch at the NCI for many years, and his role there was as the gatekeeper and the curator for the natural products library. And this is a fantastic resource of some 400,000 extracts that people in drug discovery and drug development use throughout the country. And there's probably at least 100 different academic labs and biotech and pharmaceutical companies that use this resource. And secondly, he and Gordon Craig frequently publish a review article on the bioactive natural products, in particular the ones that are active in cancer. These are some of the most highly cited review articles in the field of natural products chemistry, and they're a resource that all of us use to really assess the literature and try to make our NIH grants as strong as possible. And today he's going to speak on a topic that I think is very important, which is the role that endophytes play in natural products. And I think this is an area that will actually very likely change or revolutionize how we look at natural products from macroorganisms in general. So please join me in a very warm welcome for David Newman.
Thank you, Mark. I'm glad that he didn't list a lot more of my more dubious bits of knowledge. Luckily, he doesn't know them, though had Gordon been introducing me, let's say you would probably have run from the audience by now. One other nice thing, I did notice that the tomatoes were only cherry tomatoes so that rotten tomatoes will not reach me. Because by the time I finish, some of the members of the audience may wish to use them. However, I should point out, as Mark did, natural products, if I can get this over there somewhere, natural products in my lexicon are pure compounds coming from any natural source. They can be land organisms, thank you. They can be any form of material. So what I'm going to do is give you a very rapid idea of where things came from, and these are some of the agents that I will talk about. But initially, I'm going to tell you a little bit about how this occurred, because I am both a card-carrying microbiologist and a card-carrying chemist. So I was working in the marine area, but looking for antibiotics. And this was when I was in industry, and a very young student came along one day, and she had a moon snail extract. And in my lab, and this was in what was then, what is now GlaxoSmithKline, but at the time was SmithKline in French, and I ran the microbial biochemistry section there. We suddenly found that this produced chloramphenicol. Now wait a moment, there's something wrong here. Because chloramphenicol is a very well-known, though not used anymore in the US, antibiotic that comes from a terrestrial microbe. Yet this came from a moon snail out of the Gulf of Maine, and also the Bay of Fundy. Well, there are no sewage plants handy, which would be one potential source. I did not believe it, so I went round this young lady's college, and they'd never had chloramphenicol. I reordered the material, and sure enough, chloramphenicol was present in the moon snail. Well, I didn't think much about it until the following year. An old friend of mine, very well-known marine natural products chemist who died a few years ago, John Faulkner, discovered from a sponge collected off the west coast of California. There is only the west coast of California. I normally say the Eastern Pacific, and people wonder where that is, because everybody knows of the South Pacific or the Western Pacific, but the Eastern Pacific is, 200 nautical miles off the California coast. And John discovered, or his graduate student discovered, the Rhaneriomycins. Well, these are structurally, except for this part, identical to well-known anti-tumor agents, the saffromycins, which came from terrestrial microbes out of Europe. Again, wait a moment, there's something wrong here. You can't have a marine organism and a terrestrial antibiotic or anti-tumor agent. So one thought. Well, I recognize this particular part of the molecule as a compound that we'd worked on extensively, and it's still emblazoned in my mind. BC1794 was the name of the organism that we'd worked on at SmithKline, and it's part of the naphthyridinomycin family. So here you have a bacterial product in a sponge. Well, needless to say, John and I were not persona grata in a lot of the marine natural product areas because we dared to go against current dogma. Well, just to make it a little better, in Palau, and I had the dubious distinction when I was chief of the branch, I had to go to Palau regularly. It was a horrible job. I had to go in December and January, and it was paid for by Uncle Sam. Thank you, taxpayers. And of course, the site of the contract was 30 meters down in the blue hole at Palau, and I do happen to have a dive certification, so I had to visit. However, a very, very bright graduate student of John's, actually two of them, later on, this is the yellow sponge that you will see, as a yellow internal sponge that you will see coming up a few times. These two agents were found in that sponge. And they located those agents, and this, by the way, was in 1998. Technology was not there. But they came from basically the bacterial area of the sponge. Well, what happened next? This is where it gets even wilder. This compound, if you just take this piece, is pederin. Now, for those of you who know any Brazilian work, pederin is the blister beetle toxin. So now you have a scuba diving blister beetle operating, in this particular case, off the shores of Okinawa, producing a compound that had only ever been known prior to that time from a blister beetle, because this piece is basically nothing more than amino acid with a fatty chain. Well, suffice to say that what happened was an extremely talented young German biochemist or chemist and a German entomologist happened to be talking one time. The German entomologist was working with the blister beetle, Piderus, but his, instead of coming out of Brazil, came out of the area where Iraq, Turkey, and Iran come together. And in that area, they discovered that there were two nests of Piderus, one with a little bit of toxin and one with a lot of toxin. And it's only the female of the species, bear this in mind, gentlemen, it's the female of the species that are the worst. This was definitely the case because the female of the species was the only one producing pederin. However, when they looked at it, and for some reason they put it under a dissecting scope, discovered that there was a pseudomonal contaminant, so they thought. And, would you believe, the low-producing toxin, little pseudomonad, the high-producing toxin, a lot of pseudomonad. So they took the pseudomonad, and Jörn went in and did a genetic analysis. And what he found was the genes producing pederin were in that particular organism. So he then proceeded to present this work at a Society of Industrial Microbiology meeting in 2002. But one of the slides he put up later on was this, but without the title onomite, because he didn't know what it was. But it was one that could come from the genes that he'd pulled out. So over a beer, I suggested he get in contact with a chap in Japan, Shigeki Matsunaga, and a group of us who were at Sea Farm at one time. pre my days at NCI, who'd worked on a close relative, the mycalamides, and onamide. And sure enough, the genes from the Pseudomonas epiphyte of the toxin-producing beetle lit up the genes in the sponge that produced onamide. To cut a very long story short, he took, and this is a Herculean task, he actually took 8 million fractions from a total breakup of the sponge and proceeded to see if he could find anything. Well, what he found was this currently as yet uncultured microbe. Now, these are single, but they look as though they're filamentous, but it's actually single microbes. And there are two variants on this. They differ very slightly. And this was done by taking one cell, expanding to get the total genome, and then proceeding to do the analysis. And it turns out that 31 of the 32 then known bioactive compounds from that sponge come from that particular microbe. So there you have absolute and utter proof that it's not the sponge that produced the molecule. It's that particular microbe. So today, I'm rash enough to say that unless, if you're looking at the marine environment, unless you can show the microbe that it came from, don't even bother to publish it. So it's about, I'm going to say 80% of the materials that you see from the marine environment, and there are about 25,000 of them, I might add, pure natural products, probably 80% of them are either from free-living bacteria or bacteria that are not yet being cultured. Now, just to show you the size of the situation, Rita Colwell, before she was director of NSF, took a slide from the Chesapeake Bay, she was at the University of Maryland, proceeded to feed it nalidixic acid, which stopped all bacterial reproduction, and then did a DAPI stain. She could count over 1,000 different microbes in that stained material. She could culture 2. And as it stands at the moment, probably better than 99% of all microbes found in the environment have not yet been capable of culture. So when you and then if you look at it another way, if I asked anybody, how human are you? The answer is between 10 and 15%. The rest of you are bacteria and fungi and protists. And of course, that applies to every other organism. So let us now move to plants. Oh, sorry, 2 more. This is almost moving into the plants. It so happens that lichens also contain the genes that produce pederin. So now you're moving from a blister beetle via a sponge to a lichen. And it's actually a cyanophyte that produces it. But what happens here is that it produces a truncated version of pederin. It's not ring-closed. So that particular gene has gone everywhere. And at the moment, there are 36, plus these 2, 38 known compounds that are generated by that particular set of microbes. That's in the pederin series, plus the 30 others that have so far been isolated from Theanella swinoi. So now, does this mean that plants have a producer in their endophytes in their back pockets? I'm being a little sarcastic, but it's probably true. I think we're at the stage with plants today where we were with microbes in the 1980s. That is, there's evidence coming up now that says that endophytes and epiphytes are involved in the production of a significant number of molecules that are thought to be plant only. So let's continue. The endophyte theory, and this is from an extremely talented young Indian chap who just got his habilitation at the University of Dortmund, Suvik, he's extremely talented. This is merely, I'm not going to go through it, it simply says that an epiphyte on a leaf or in a leaf or somewhere else in the plant, you've got feedback loops from insects that are attacking, fungi that are attacking, bacteria that are attacking, and then you've got a feedback loop that says produce and defend. And if you think about it, it actually makes sense. So now, if you look, there have been a significant number of compounds of interest to me, and bear in mind I was NCI, so though I did 20 years looking for antibiotics, I did 25 years looking for anti-tumor agents, so I have a certain bias. But it turns out that there are a significant number of well-known plant-associated, being careful now, agents that are clinically used in cancer. And I put 3 of them up here. And in fact, Taxol came directly from my branch, but it was before I was chief. This became a drug in 1992. It was approved by the FDA. It was isolated from the bark of the taxus tree, a trash tree, and the joke at the time was, and it was a rather bad joke, every woman should go out and plant 6 yew trees in her backyard because you required 150 centimeter diameter yew tree per treatment to get enough of the Taxol. And it turns out that that's not the case anymore. But Taxol was one. Prior to the work on Taxol, the same people who did Taxol had also worked on camptothecin. And we actually gave these extracts to academics to work on in an earlier iteration of the natural products branch. So Mansukh and Wani did camptothecin, and Wani and Mansukh did Taxol. So here are 2 gentlemen who have 2 of the most famous plant-associated anti-tumor agents done by them, but from materials collected by NCI, and then we commissioned academics to work on them. The third one, podophyllitoxin, actually has an ethnobotanical background in that the plant was used by native Indians for the treatment of genital warts, which is in fact a version of cancer. So podophyllitoxin, camptothecin, and Taxol were reported by a variety of plant organisms and organisms from plants to produce these on fermentation. But each time they were subcultured, they lost the activity. So you started off with 100% production, and then it went down and down and down until you needed extremely good techniques to find them. However, very recently, this came from a Vietnamese medicinal plant, and this was a group funded by NCI, and they found this agent. This was published only a week ago. Well, that's podophyllitoxin with a couple of pieces added to it. So is it bacterial or fungal in origin? The answer is probably yes. If you take the Taxol microbe axis, this is work came from Kusari and others. They found that there is, in fact, feedback loops. There's an endophyte that does, in fact, produce Taxol. It has the complete genetic machinery for producing Taxol. Its taxodiene synthase, which is the essential part of the mechanism, differs from that of the plant. So this is not a carryover, nor is it genetic transfer. This is genuinely a microbial Taxol. And it's been totally sequenced and shows the activity. There are questions, and these are relevant questions. One of the major problems is what do you do next? Well, you can remove the genes, which have been done, from the fungus, and if you're capable of doing it, you can end up with a way of producing them by using fermentation techniques. However, the major problem were yields. And this is something where academics are at a disadvantage. The reason being that a very large amount of the work on yields, increasing yields of bacteria and fungi, was done in industry. And it was done, now I'm 78, it was done when I was in my 20s. And so this work was not published. But if you ever worked in the biochemistry area or in the microbial biochemistry area of any of the big pharmaceutical houses worldwide, there were techniques known, never published, but there were techniques known to induce production of molecules that you had of interest. We used to call it putting everything, including the kitchen sink, into the fermentation. Academics have only just rediscovered this within the last few years. But it's a very well-known process. But because Big Pharma worldwide got out of antibiotics by fermentation, all that experience, because we never wrote it down, that experience is basically gone. However, it's being rediscovered. So then, if you just take this was a precursor treatment with a camptothecin producer, where they put in some of the precursors of camptothecin and got increased yield, which would be expected. This was a system that we used, and I was describing this to some of Mark Hammond's students yesterday evening. There's a device known as the Ecologen, which I was using back in the 1960s, where you are able to mix and match microbes. And I subsequently did some more work on this, and a lot of people have done work on this now, whereby you take two dissimilar microbes and ferment them together. And what you get? Something you've never seen before. You get expression of cryptic clusters, you get increased expression of one, and this is a technique that works. Well, this is an example where people took two fungi from a camptothecin-producing plant, put them together, and you got an increased yield of camptothecin. Obvious to me, not so obvious to others, because the thing you have to remember is that a plant is not an axenic container. Anything that's used to produce a plant product, in quotes, by means of cell culture, it's an aseptic process, yes, but the plant culture, the callus culture, is not axenic. There's a very simple test that nobody, to my knowledge, has ever published, where you simply take a microtome through a producing callus culture and stain it for the number of nuclei per cell. If you find more than one, you've got endophytes in there. This was done, and the mixed culture produced increased yields. This was a beautiful piece of work done in conjunction, I might add. It's Nick Oberle's at Greensboro in North Carolina. Nick works hand in glove with an old friend of mine who's an expert chemist, but has a collection of about 70,000 fungal isolates. And Nick is slowly working his way through them. But because Cedric Pierce, who runs that operation, Cedric knows how to persuade bugs to produce. What Nick did, his student did, they took the milk thistle, looking for sibilants, and isolated fungi. When cultured, they produced the sibilants. If you subcultured, you got no production. If, on the other hand, you subcultured, again, from this non-producing culture, and you took a wash of the leaves of the milk thistle, and you filter sterilized that wash and added it back to the fermentation, lo and behold, they're produced again. So there's something in the leaf, on the leaf, or with the leaf that is inducing production. So yes, this is a plant-derived product, and it's probable that there's talk between organisms producing sibilants when necessary. And this was just done again to show the same thing. These are ways in which you can take that sort of data and produce or increase yields of materials. Now, rohutakine, for the chaps from India amongst us, this was Herxed India, work done approximately 30 years ago from a plant that produced rohutakine. This led to two drugs in clinical trials, including this one, flavopiridol, which NCI actually took into phase 3 clinical trials. It turned out not to be as efficacious as we thought, but it did give us an awful lot of information that people didn't realize about how molecules of this type at nanomolar concentrations would activate or deactivate cell cycles in cells. So this is an example where the base molecule was then modified slightly to give better pharmacologic properties. This is merely, I can jump this slide. Swansonine, for those of you who, and Mark will remember this because it's loco weed, Swansonine is a product of a fungus that is carried in the seed. So I always joke, because I'm old enough to remember Lamarckism, that this is Lamarckism because it's an induced trait that's carried on by genetics. I'm joking about the Lamarckism, but rather than Mendelian genetics, but still, it's the same thing. If you take this seed where swansonine is produced and you treat it with a fungicide, you don't get swansonine, but you still get these, which come from the plant. So these are probably genuine plant products, not seed products, not products of an endophyte or an epiphyte, because there's nothing there after you've treated the seed. This one, golden seal. Again, now this is Pierce and Oberle's again, with assorted others. Golden seal, one of its major uses is as an antibiotic or as an anti-infective agent. Well, pulling the fungi off golden seal and out of golden seal, you end up with this rather simple molecule that happens to be a rather potent antibiotic and is possibly the reason for the anti-infective use of golden seal, or one of the uses. This is another one, and now this is from traditional Chinese medicine, where the boxwood, this is the fungus from it, these are the molecules from the fungus, and they are anti-infective agents. So again, this is an example of where the product from the plant and the product from the fungus are the same. Now, this is one that's lovely, because those of you who remember red rice or remember mevinolin or lovastatin, which was the first of the statins to go into clinical use and was in fact the natural product, even though it was produced by fermentation, it was from the fungus. Well, taking the traditional Chinese medication here, when this was, Ayurvedic medication, it was fermented and produced lovastatin. And actually the yield from the plant is roughly the same as you get from taking the producing agent of lovastatin from a culture collection and fermenting it under the same conditions. So any cholesterol-reducing activity from this particular organism is due to the lovastatin produced by the microbe, the fungus that is in there. This is another example, podophyllitoxin, but now camphorol, from a fungus isolated from the plant that produces podophyllitoxin. So you have at least two genetic systems running here. And one asks the question, how many more? Homoharentonin, well known in traditional Chinese medicine as a treatment for leukemia, and was approved in the US in 2012. Well, there are a series of papers, and because my Chinese is non-existent, I should say my Mandarin is non-existent, I was not able to get a translation of that paper, but one of my colleagues did translate it, and yes, there is a fungus that produces homoharentonin, and that fungus was isolated from the plant that homoharentonin was originally isolated from. If you go to maytansine, now this went into clinical trials in both Europe and the United States. Council Maytanus tree, originally Eastern Africa, Ethiopia. It was when the structure of this was first seen, except for the ester here, that people with my sort of background said, wait a moment, that's ansomytosin P3. That's not a plant product. That's a microbial product, a streptomycete product that's been known for many years. It turns out that they were absolutely correct. What has happened now is that maytansine is produced as ansomytosin P3 as part of the rhizosome of the plant another bacterium as yet unidentified, but it is a prokaryote, in the same rhizosphere, converts it to maytansine. The plant then takes this up as a protective agent, as an antifungal agent, which basically is actually the same thing with Taxol. So there's absolutely no question maytansine is not a plant product. It is a microbial product that's been adopted by the plant. And I might add by other plants, mosses and so on and so forth. It's found in a lot of places around the world. Oops. So terpene synthases. Now, anybody who teaches has always been told that terpenes are products of the eukaryota. Oh no, they're actually products. There are well over 250 known, identified materials from Streptomyces that produce terpenes. So now the question comes, where do your essential oils come from? Because they're all terpenes. My answer is probably from a Streptomyces and or other bacterium, not a fungus, and not a eukaryote. My final slide. And if you can't see them, they're hand grenades, but the pin hasn't been pulled yet.
We do have time for one very good question.
Well, you see, if we'd cut Mark off earlier, we could have had time. We could have had time for not this Mark, t'other Mark. We could have had time for more questions. Anybody foolish enough? Throw a hand grenade? Go on, Mark.
So do you have any, or can you comment on any possible metabolites that are bona fide plant-derived metabolites?
I would say that the examples that I gave with a few slides back, with the loco weed, they probably are. There's a question, to my mind, of even the vincas, because there are reports out of China of microbes producing vinca alkaloids, and there are now reports from Nancy Keller's group showing vinca-like materials produced in isolated fungi. I think we got Jim. We got to allow Jim.
We do?
Of course. Hey, come on. We used to fund it. Got to allow Jim.
But you don't anymore.
Well, he still gets funding.
What proof is there that terpenes, for example?
I don't know of any that says they're mutually exclusive, but I do know that with the what would be a very interesting thing to do would be to take a plant that has the essential oils, look for the genetic production of those, because with the terpene synthases, they're well known, and see what you find. That would be my answer, Jim. Yes, it's possible. But I suspect, seeing that microbes were around at least 1 billion years before plants, that I think it may be from the microbe to the plant rather than the plant to the microbe. So it's just to make you guys think in a slightly different way. Okay?
So the pleasure of this conference is we'll all be together for quite a while longer. So again, I hope this crosstalk continues. Thank you very much, Dave.
Pleasure.